Hello, everybody. We're going to keep talking about cellular metabolism. And in the previous lecture, we talked about the importance of doing something with pyruvate. So, glucose is one of our stored energy macronutrients, and it can undergo metabolism without the use of oxygen through the process of glycolysis. The end product of glycolysis is pyruvate. We discussed this problem that if you don't do something with the pyruvate, it will go through end product inhibition and actually stop glycolysis. So in the previous lecture, we really went into detail about what happens to pyruvate when there is insufficient amounts of oxygen available. And when there's not enough oxygen, we can convert it to lactate. And we talked about the benefits of being able to produce lactate and what that does to help facilitate cellular energy metabolism. This lecture is all going to focus on the role of oxygen. So we finally get to talk about what oxygen does in this story after all semester telling you how important it is for the lungs to bring in oxygen, how important it is for the heart to pump oxygen, how important it is for the blood vessel to deliver oxygen. Let's finally talk about what in the heck we do with that oxygen. So as it says on the top of your notes, there is another enzyme that can act on pyruvate, and that is pyruvate dehydrogenase. But first, we have to get the pyruvate into the mitochondria. So I'm just going to draw kind of a big old mitochondria on my screen. This is our mitochondria. And we're actually going to transport that pyruvate all the way into the mitochondria matrix. So now that pyruvate is inside of the mitochondria, the enzyme, what color should we use here? The enzyme pyruvate dehydrogenase. Again, these names of these enzymes, very cool names, something to consider. Pyruvate dehydrogenase can convert this 3-carbon pyruvate molecule into acetyl-CoA. Oops, forgot the L. Acetyl-CoA. It's a little dash there. The conversion of pyruvate to acetyl-CoA does strip an additional electron off of that 3-carbon molecule. So in biochemistry, if we need to move a molecule, we are going to need an oxidizing agent to accept it, because we can't just throw electrons around. They're very reactive. So this enzyme of pyruvate dehydrogenase does require an NAD as an oxidizing agent to accept a hydrogen, because hydrogen is how we move electrons in biochemistry. So pyruvate dehydrogenase is going to require an NAD to accept an electron. We are actually also going to produce, for the very first time, a carbon dioxide. This is an important one to note. We talk a lot about how carbon dioxide is a waste product of metabolism, and so far we have not, in any of the processes of our phosphagen system, of glycolysis, we have never produced this key waste product. We're always talking about blowing off CO2. This is the first time we see CO2 enter the scene. So where pyruvate was a 3-carbon molecule, we have now lost one of those carbons. Here it is. It's the carbon of carbon dioxide. So acetyl-CoA is a 2-carbon molecule. We have blown off one of our carbons. Let's look at this same story in more of a textbook figure, if that works better for you than my drawings. Here is the pyruvate that was formed out in the cytosol through the process of glycolysis. The yellow on this figure is all of the mitochondria here. So we have to get the pyruvate in. Here's the transport protein that allows pyruvate to enter into the mitochondria. And then through the action of pyruvate dehydrogenase, the enzyme, we are able to convert this 3-carbon pyruvate into the 2-carbon acetyl-CoA. One of the carbons that gets lost is out here, produced as CO2, and we also strip some electrons off and load up an NAD with some electrons in the form of hydrogen. So at this point, we are inside of the mitochondria. We have formed acetyl-CoA. There's one more important thing about this step. This step is irreversible. Ooh, I can't spell irreversible. It doesn't go the other way. This arrow points one way. Remember, lactate dehydrogenase was a reversible step. You can make lactate, and then as soon as you have enough oxygen, you can just, boom, pop it right back, make it pyruvate again, and keep that sucker going. Once pyruvate has been converted into acetyl-CoA, it doesn't go back. So this is an irreversible step. We can't change our minds. We are stuck with this 2-carbon molecule in the mitochondria. If we look down at the next part of your notes, it's now describing the citric acid cycle. This cycle is also called Krebs cycle or TCA cycle, if those are the names that when you first heard about this pathway. So this is, again, we're within the mitochondria, and this is a really important step in the metabolism of glucose. And let's start in the very top corner. Up here, we can see this is the step that we've already described. So technically, this conversion of pyruvate to acetyl-CoA is not part of the citric acid cycle, but a lot of times we just kind of lump it in there because it creates the first product that enters this wheel, acetyl-CoA. So this step has already been discussed. You'll see the full cycle in your notes. And I do list all of the enzymes because I think it's important to know that between each of these products of the citric acid cycle is a specific enzyme that makes the change in the conversion of that molecule that is necessary for the TCA cycle to keep going and be a continuous loop. It is not, I don't think, necessary for a physical therapist to know the names of these enzymes. So I'm certainly not going to have you memorize the names and all the words of the TCA cycle. The story of what it does, however, is really important. So I included it in your notes I kind of made the enzymes a very light font so that you wouldn't be tempted to memorize them. I know you guys are so tempted to memorize extra things that you don't have to, but fight the urge. If it helps you, if you're a real strong chemistry person and you need those names and kind of that understand the enzymes to learn this pathway, please have them there for your reference. But you will not be expected to know them. So I'm going to simplify this figure and just take the enzymes out because what happens in this wheel is important. So the very first step of the citric acid cycle is acetyl-CoA joins with oxaloacetate. And this oxaloacetate, or OAA, is an important component of the citric acid cycle to know. And it will come into play, especially when we start talking about ketogenesis. So if acetyl-CoA shows up and it's in the mitochondrial matrix and there is an oxaloacetate available, we can travel around this ring. Acetyl-CoA and oxaloacetate can combine and be formed into a citrate. And around it goes. Each enzyme will then do its job to slightly change and convert this molecule until we ultimately end up back making OAA again. And when OAA is made at the end of this cycle, if there is an acetyl-CoA waiting for it, we go all the way around the loop again. So the citric acid cycle, the first step is the joining of acetyl-CoA with oxaloacetate. And then if you go all the way around, the final step makes an oxaloacetate, which is why it is a cycle or a circle, because the final product is also the first step. Things that are made along the way is important. And this is not in your picture on your notes because I want you to add it as we talk about it. I'm putting these additions on my picture at the actual steps where they happen. I will never ask you where it happens. Like, is it the conversion of isocitrate to alpha-ketoglutarate? Or is it fumarate to malate? I'm not going to ask you that. I don't care that you know exactly which step it happens, but the products of the TCA cycle play a role in our overall metabolism. So let's add them to your figure, okay? The first thing that we get, let me just erase my markings on here so they're not in the way. The first thing that we get is we load up some of our electron carriers. There's actually 3 distinct steps where we need NAD available to strip electrons off of the molecule and load them up and as an NADH. So you can add these to your figure, but make sure that you add 3 separate steps where NAD is required, and you end up yielding 3 NADH, which is also frequently written as H plus kind of hanging around, if that confuses anyone. I mentioned at the beginning that we often talk about this pyruvate conversion to acetyl-CoA step in the conversation about the citric acid cycle. So I'm just going to, on this slide, kind of include that as part of this discussion because it'll play a role in the next step. So let's remember that before we enter the TCA cycle, there was this conversion of pyruvate to acetyl-CoA. So there was pyruvate oxidation as we stripped an electron off at that step as well. So as it's written above in your notes, just above the TCA cycle, the pyruvate oxidation gave us 1 NAD, and the citric acid cycle gave us 3. We actually now also get to talk about that other electron carrier. So far, the only carrier on the scene has been NAD, but in our discussion on oxidation reduction reactions, we talked about 2 main carriers, NAD and FAD. And in the TCA cycle, one step down here at the bottom does take an FAD and load it up with an electron. So the citric acid cycle yields 3 NADH and 1 FADH2, 4 separate high energy electron carriers. The next thing that we get from all of this work, all these steps of changing and moving things and going around the bend, we get 1, that's right, 1 high energy energy donor, a GTP, a high energy universal energy donor. It has those phosphate bonds like an ATP that makes energy. So when we discussed ATP, we said adenosine triphosphate is, the primary universal energy donor. This step technically makes a GTP. You can think of this as an ATP. It is another universal energy donor. So the TCA cycle only gives us 1 molecule, only 1 in all this work. The other thing we get by going around the cycle is we strip off additional carbons. There are 2 steps that produce carbon dioxide. So if we add to our list of what we get from the TCA cycle, we get 2 carbon dioxide. I like having that first step of pyruvate to acetyl-CoA on this slide because that is another step that produces carbon dioxide just prior to the TCA cycle. So starting with 1 pyruvate entering the mitochondria, that 1 pyruvate, if it goes from pyruvate to acetyl-CoA and then all the way around the TCA cycle, produces 3 total carbon dioxide. Now, I hate to blow your mind, over a computer screen, and it's much more fun to just watch the gasps of awe when it's in, 356, but you're going to have to gasp in awe in the privacy of your own home. Here comes, are you ready? In all of cellular energy metabolism, this is the only place we make CO2. Oh my gosh, I can almost hear the gasps from my office as I record this months before you listen to it. This is the only place we make carbon dioxide. Let's go back to that first description of the metabolism of glucose. Do you remember we took some time to write this out? And we said that when you start with a glucose molecule and it goes through complete combustion, it ends up becoming water and carbon dioxide. So we know so far that if we take a glucose molecule and send it through glycolysis, so this dotted line is glycolysis, we end up with how many pyruvate? We end up with 2, right? We end up with 2 pyruvate. When pyruvate goes, maybe I should draw this as the circle that it is. When pyruvate goes all the way through the TCA cycle, we end up making 3 CO2 per pyruvate. We started with 2. So the total production of carbon dioxide, the 6 CO2, we have now accounted for. So by starting with glucose, 1 glucose molecule becomes 2 pyruvate. Each pyruvate that goes through the TCA cycle makes 3 CO2. So we have seen where all of the carbon dioxide comes from. 1 from the conversion of pyruvate to acetyl-CoA, and 2 per turn of the TCA cycle. So we start with the glucose. We have broken it down, and what are we left with? A little teeny tiny bit of energy, electrons loaded up in their trucks, and carbon dioxide. I like looking at it like this. The steps that we've done so far, we started with glucose, and almost at each step along the way, we stripped an electron off, loaded it up in a truck. Stripped an electron off, loaded it up in a truck, step by step by step. First out in the cytosol through the action of glycolytic enzymes, then through the enzymes of the TCA cycle, and what are we left with? Nothing but a waste product to go breathe off in our lungs. But we haven't really made the energy yet. We only got 1 little GTP from all that work. We barely got any ATP from glycolysis. It was okay, but not nearly enough to power human movement and this amazing ability we have to do exercise. Each of these loaded trucks needs to be delivered to the electron transport chain. This is really where the magic happens. I know I keep claiming the magic has happened, but now we're really about to get into the magic, because those electrons carry a lot of energy with them, potential energy, that we can harness through the electron transport chain and pump out massive amounts of ATP. So let's just first remember where we are and what we're talking about. At the top of your notes, you get the term oxidative phosphorylation. This is what we call the process of ATP generation powered by the transfer of electrons from those high energy carriers, NADH and FADH2, ultimately to oxygen. So what we need to do is transfer these electrons carried on our flatbed trucks and deliver them to oxygen, and through that process make ATP. That is oxidative phosphorylation. So this all happens in the mitochondria, and there are steps that really are important to remember where we are in the mitochondria. So I have a little space for you to draw this out with me in your notes. So this squiggly looking picture in your notes is a mitochondria, and I just want to review the structure of a mitochondria. We have this outer membrane, which is a phospholipid bilayer membrane. There's many proteins and channels that allow the transport of molecules and ions in and out of the mitochondria. Then there is this inner membrane, a second membrane that kind of is this squiggly line within. And it separates the mitochondria into two distinct spaces. You have between the inner and outer membrane, very appropriately named, the intermembranous space. And that is the area between the inner and outer membrane of the mitochondria. And this center area that the inner mitochondrial membrane is wrapped around is the matrix. This is the mitochondrial matrix. So we already talked about the mitochondrial matrix. This is where the enzyme pyruvate dehydrogenase lives. Remember, we delivered pyruvate into the mitochondrial matrix for that first step of converting it to acetyl-CoA. This is where that happened. We transported it all the way into the mitochondrial matrix. This is where acetyl-CoA joins with oxaloacetate, and we have that TCA cycle. There it is, kind of tiny TCA cycle. It's good to remember where we are. So on the inner membrane is the site of the electron transport chain. The inner membrane is the site of the electron transport chain. The inner membrane contains these 5 complexes of integral membrane proteins. So I'm going to draw them crossing across the inner membrane, and I'm going to label them 1 through 5. The 5 integral membrane proteins of the electron transport chain. So within the mitochondrial matrix is where the TCA cycle just produced all of those high energy electron carriers. And here along the inner membrane are the proteins of the electron transport chain. This first protein gets its own name. This is NADH dehydrogenase. They all have names, but these are the ones I'm going to care about. And the fifth is going to be a huge role. This is our ATP synthase enzyme. So we'll talk more in detail about those. So this is the site of oxidative phosphorylation. And it's good to just think about what that word means, oxidative phosphorylation. If I have space up here on the top. Am I spelling that right? Get it out and then check. Phosphorylation. Hey, it came out right. All right, oxidative phosphorylation. It's amazing how much my hand moves and my brain's thinking about something else when I'm spelling. It's dangerous. Okay, what does oxidative mean? Oxidative is a loss, right? So oxidative phosphorylation is the removal of electrons, and then phosphorylation is when you add a phosphate. So the process of oxidative phosphorylation is going to remove electrons and ultimately add a phosphate. Remember that phosphate is going to be those high energy bonds. So keep that in mind as we go through our story. So a little summary piece right in the middle of your notes to keep this in the context of our story. We started with glucose out in the cytosol. Through glycolysis, we did get some ATP, and we got some energy carriers driving over to the electron transport chain. Specifically, from glucose metabolism, we ultimately get 10 NADH. 2 of them are from glycolysis. 2 are from pyruvate to acetyl-CoA. The only reason that there is 2 here is because for every 1 glucose, we get 2 pyruvate. And every time pyruvate is turned to acetyl-CoA, we generate an NADH. And because there's 2 of them, That's where the 2 comes from, because we're talking about starting from one glucose molecule. So for every 1 glucose molecule, we get 2 NADH from glycolysis, 2 from the conversion of the 2-pyruvate to acetyl-CoA, and then 6 from the TCA cycle, because you get to go around it 2 times because you started with 2-pyruvate. You also get 2 FADH2, which is, again, because you were able to go around twice. So we have 10 NADH, 2 FADH2 loaded and ready to be delivered to the electron transport chain. This is one more visual of the mitochondria. If you can use this better than my drawings, here we have the outer membrane. So out here is the cytosol, the intermembranous space, and then the inner mitochondrial membrane. And you can see embedded in that membrane are these proteins of the citric acid cycle. In the center matrix is where the TCA cycle is pumping out these carriers of electrons, ready to deliver it to the electron transport chain. I'm going to draw this out in that space you have in your notes. I encourage you to draw along with me. Let's just orient ourselves first. I'm going to make this green line the outer membrane, so that out here is cytosol of the cell. And I guess the same colors, okay. And then we're going to make this guy here the inner mitochondrial membrane. And I'm just going to give that look of the phospholipid bilayer, just to remind us that it is this membrane that proteins can kind of get embedded in. So if we've got the outer membrane at the bottom and the inner membrane kind of cutting across that space, your screen, that means that this is the intermembranous space. Kind of all of this is intermembranous space. And on the opposite side up here is going to be the matrix. So is everybody oriented with me? Here we go. This is kind of the outer wall of the membrane of the mitochondria. Let's embed the enzymes of, or the membrane proteins of the electron transport chain. All right, so here is 1. Here is 2. Then we got 3. Spanning the inner mitochondrial membrane. Then we get 4. And then 5, I'm going to draw really funky because this thing is so cool. It looks like this. This is number 5. And number 5 actually gets its own name. It's called the FOF1. And this is the same as the ATP synthase. Just a couple names. We'll talk about that as we get to that step. So within the mitochondrial matrix is where we are producing the NADH and the FADH2, pumping it out from that TCA cycle, going round and around, and NADH gets delivered to that very first membrane, integral membrane protein of the electron transport chain, which is the NAD dehydrogenase. And that name, NADH dehydrogenase, means it can rip that hydrogen off. So when NADH shows up at this first step of the electron transport chain, that enzyme rips the hydrogen off and re-releases the empty NAD. It recycles it. And this NAD is then free to go to the TCA cycle and accept another electron. And around we go. So as long as NADH is able to deliver its hydrogen to that very first complex of the electron transport chain, we can keep on recycling NAD and have open flatbed trucks available to accept electrons from the TCA cycle. Now, what this first step of the electron transport does is very unique. So pay close attention because I'm about to kind of throw you a curveball. This entire time we have said that in biochemistry, electrons are carried on hydrogen. That's because hydrogen has that one electron circulating around it in its orbit, and it's a perfect carrier of electrons for that reason. At this stage, complex 1 takes that hydrogen and it rips its electron off. So now we actually have a free electron. It takes the hydrogen and it rips the electron off and it spits out a proton. So notice the positive charge here. This is not a hydrogen, that's just a simple proton. It's been divorced of its electron. So step 1 of the electron transport chain rips this hydrogen off of the NAD. And when that hydrogen goes into the complex is able to trap the electron, rip it off of that hydrogen, and spit out a proton. So it would be wrong to call this guy down here now a hydrogen because it's not really, because it doesn't have an electron. At this point, you can think of the electron transport chain as a game of hot potato. And the potato is the electron. So once that electron has been ripped off of the hydrogen and is trapped in complex 1, it's going to toss that electron to complex 2. Complex 2 is going to use some of the energy of that electron to pump another proton into the intermembranous space. And 2 is going to toss that electron to complex 3, which is going to use the energy of that electron to pump a proton into the intermembranous space. Complex 4 is going to get handed the hot potato, the electron, and it's going to use that energy to pump a hydrogen into that intermembranous space. So NADH shows up at complex 1. That enzyme rips the hydrogen off, traps the electron, spits the proton off, and then is like, "Hoo, hot, hot, complex 2, you take it." Complex 2 grabs the electron, "Hoo, hot, hot, hot," grabs a proton, shovels it through, and passes the electron on to complex 3. Complex 3, "Ooh, this electron, so hot, ow, ow, ow," grab the proton, pump it through, pass it to complex 4. Complex 4, same thing, but something else very magical happens at complex 4. Put a pin in that. What has happened to the electrons as we've gone along this chain? Every time the complex takes that electron, it's going to move the electron to a slightly lower energy state. It loses energy because there's energy that is being used by the electron to pump the proton across the membrane. So as the electron passes along the chain, it moves to a slightly lower and lower energy state. Complex 4, this last complex, it has the electron, and this is where we are going to be able to reduce an oxygen molecule. The last step, complex 4. I'm just going to go to a new screen to draw this out, and I'll go back and add this to the picture. I just don't want to muddy the picture. This last step in complex 4 is going to take 2 of its electrons and add it to 2 proteins. Stick an oxygen on there, and it's going to make water. So complex 4 of the electron transport chain is going to take that electron, combine it with protons and an oxygen, and make water. What's really important, we have finally talked about a role for oxygen. Let's go back to our picture and draw this in. So at what color should we use? Something different. At complex 4, if oxygen is available, it will take that electron out of complex 4, use a proton, and make water. Complex 4 is the only place in all of these steps that uses oxygen. So why is oxygen so important in cellular energy metabolism? If complex 4 cannot get rid of its electron, then complex 3 won't be able to pass its electron on. If complex 3 can't get rid of its electron, then complex 2 can't give it its electron. If complex 2 can't give it its electron, then complex 1 won't be able to get rid of its hot potato. If complex 1 is currently holding an electron, when the NADH shows up at the electron transport chain, it will not be able to deliver its hydrogen, or in other words, it will not be able to deliver that electron. We will not recycle NAD, and what will ultimately happen is more and more NADH will start to accumulate, all loaded up with electrons, piling up here saying, okay, we're ready, we got electrons, hot potato coming at you, and they will have nowhere to go with them. And you'll fill up all of your NAD with electrons, but not be able to do anything with it. So oxygen is so important because it is the final electron acceptor. If there is no oxygen, the whole electron transport chain grinds to a stop. Because if the complexes are currently holding electrons, they have nowhere to unload it to accept the next one. If there is oxygen available, it will accept the electron from complex 4, and the factory goes back into production. At this point, you might be saying to yourself, well, that's great, but I don't see any ATP in this story. You've told me about electrons, you've told me about oxygen. Where is the ATP? Well, I'm glad you asked. This FOF1 enzyme, or ATP synthase enzyme, is the final enzyme of oxidative phosphorylation. And how it works is extremely cool. At this point in the story, we have passed the electron along the chain. And look what's happened in the intermembranous space. We have a whole bunch of protons collecting in the intramembranous space, and we have almost none in the matrix. We have created a huge concentration gradient of protons in the intramembranous space. So the final enzyme of oxidative phosphorylation, the FOF1, uses the stored energy of the electrochemical gradient to drive the machinery of FOF1. And you can imagine FOF1 as like a huge turbine at a hydroelectric plant. This entire top of this enzyme spins on its head. And the energy to make it spin is this huge concentration gradient of all of these protons shooting through the FOF1, spinning it as it goes. So all of these protons that have collected in the intermembranous space over time fuels the final enzyme, because this huge electrochemical gradient drives through FOF1, spinning it, and every time the FOF1 spins around, it takes an ADP, P, let's make that cleaner. It takes an ADP and a free inorganic phosphate, and it slaps them together and makes ATP. The spinning of this big enzyme at the end creates the potential energy, the energy to fuel this process to stick the phosphate back onto the ATP. Now we have the energy donor ready to go to fuel mechanical work. So, does the formation of ATP directly use oxygen? No. FOF1 is not an enzyme that requires oxygen. But without oxygen, there would be no proton gradient. And it's the proton gradient that fuels the FOF1 enzyme. Because the rushing of protons through the FOF1 spins this enzyme around, and every time it spins, it sticks a phosphate on an ADP and pumps out ATP. So this is a textbook image of the same process. And on the top of the next page, I have kind of the words of this story written out for you in your notes. I know this is a little bit of a confusing pathway, and I think it's challenging when I can't be dancing around in front of the room showing how this stuff moves. So read through the language in your notes, and if you find it helpful, this picture shows this exact concept again, where the mitochondrial matrix is at the bottom of the screen, the inner mitochondrial membrane is cutting across the middle, and the top is that intramembranous space. You can see complex 1, 2, kind of hidden, 3, and 4. NADH shows up at complex 1, and as it travels through the complexes, you can see the electron being passed from complex to complex, and at each stage that uses the energy to pump the hydrogen through until you get this big proton gradient within the intramembranous space, which ultimately fuels the ATP synthase, spinning it on its head as the protons fly through FOF1 and stick that phosphate onto the ADP to create ATP. This process would come to a grinding halt if at stage 4 here, when that electron showed up into complex 4, if there was no oxygen to accept that electron, ultimately making water, the electrons would be trapped within the complex, and the initial NADH would be unable to continue to dump off electrons, and this whole process would come to a halt. If we look back at our original drawing of how we break down glucose, we start with a glucose molecule, and in the presence of oxygen, it can get completely broken down. We already discussed where the 6 carbon dioxide come from. Completely, absolutely all from the conversion of pyruvate to acetyl-CoA and then the process of the TCA cycle. And now we see where the final water comes from. At the completion of the electron transport chain, we have complete combustion of the original glucose molecule. We have taken the glucose molecule and completely broken it down into water and carbon dioxide, and in the process, released and formed ATP. We got the stored energy of those original molecular bonds out through the steps of glycolysis, TCA cycle, and ultimately the electron transport chain to make the energy currency of the cell out of that glucose molecule. This is kind of a textbook cartoon of all of those processes put together. Glycolysis out in the cytoplasm, ultimately being delivered into the mitochondria. If we look at what we get, this is in the middle of your notes, the final ATP count from one molecule of glucose. We get 2 ATP from glycolysis, 2 high energy donors from the TCA cycle, and the reason you get 2 is because we end up with 2 pyruvate, so we get to go around twice. And if we went through the electron transport chain step by step by step, we would see that we would ultimately get from all of those NADH and the FADH2, we would end up with 32 ATP for a total of 36 ATP when we completely break down glucose. So at the beginning of this series of lectures, we said that there are multiple pathways that can get ATP, and they have their strengths and their weaknesses. Glycolysis is this really important pathway because it's super fast, right? It's out in the cytosol, 10-step reaction, doesn't need oxygen, but you get only 2 ATP per molecule of glucose that's going through glycolysis. To go all the way through to the end of the story, you can get a lot more ATP out, but that will take some time, and it will take oxygen. This seems like a good place to break as we've completed the story of glucose metabolism. In the next lecture, we're going to look at some of the other macronutrients that we get energy out of and make some comparisons between these pathways. Have a great day.